Swayam Prabha. Digital India. Educated India.
Hello everyone. Welcome to the next lecture of 4th unit of Plant Biotechnology course. Earlier in this unit, we learned about the plant and microbial interactions, plant growth promoting rhizobacteria, nitrogen fixation, etc. And now we are learning about the plant genetic engineering tools and plant transformation. My name is Manoj Sharma and I am working as an assistant professor of plant biology at Jawaharlal Nehru University, New Delhi. In JNU, I teach plant biotechnology and genetic engineering at School of Biotechnology. These lectures have been reviewed by Professor Kashmir Singh, who is working as a professor of plant biology at Department of Biotechnology, Punjab University, Chandigarh. And this project has been sponsored by DTH Swayamprabha MHRD, New Delhi. In the last lecture, we learned about the physical or the direct methods of plant transformation, like microinjection, electroporation, or the particle bombardment method. Now, in this lecture, I will discuss about the Agrobacterium mediated plant transformation, which is an indirect method of plant transformation. So you will learn about the mechanism of Agrobacterium infection, how Agrobacterium infect plants and transform naturally to cause the crown gall disease. And finally, how can we use Agrobacterium as a tool for plant genetic engineering? Agrobacterium mediated plant transformation is one of the most common as well as most successful plant transformation methods. Therefore, we'll talk about it in a little more detail. First, I will talk about the biology of the Agrobacterium infection and then how can we use it as a plant genetic engineering tool. Agrobacterium tumefaciens is a gram-negative soil bacterium and have quorum sensing abilities. I'm sure you would remember the concept of gram-negative, that is, these microbes do not retain the crystal violet gram strain and the presence of very thin peptidoglycan layer. Whereas in gram-positive bacteria, peptidoglycan layer is thick and they retain the gram strain. And the quorum sensing, it means that these bacteria senses the chemical signals from the environment and regulate their own gene expression in response to these signals, and it helps to regulate its growth and density as per the environmental responses. Agrobacterium tumefaciens is also called as nature's genetic engineer. That means it is capable of transforming the plants naturally. In nature, it infects the plants and causes crown gall disease. During the infection, it transforms the plant genome, that is, it inserts some of its own DNA into the plant genome which result in the development of the Crohn gall tumors. This infection of Agrobacterium that induces the production of Crohn gall tumors is called as a Crohn gall disease. In the wild, it usually infects the dicot plants and can cause severe economic losses in several crops of agriculture or the horticultural importance like tomatoes, roses, walnuts, apples, cherries, and so on. Agrobacterium tumefaciens and Agrobacterium rhizogenes are the two common species that transform plants and have been optimized for plant genetic engineering. These species harbor Ti and Ri plasmids, that is, Ti is for tumor inducing plasmid and Ri is for root inducing plasmid. In this lecture, I will focus more on the Agrobacterium tumefaciens that have a Ti plasmid and causes the Crohn gall disease. Now, let's have a quick look on the historical perspective of the Agrobacterium biology. In early 1920s or 30s, massive scale infections of the tumors were reported in the fruit orchards, like in the walnuts, apples, grapes, etc., which led to the huge economic losses. And therefore, efforts were put to identify the problem to get rid of or to take care of the disease. Soon it was identified that this disease is caused by an bacterial pathogen, and it was named as a Agrobacterium tumefaciens. During late 1930s and 40s, several developments were reported about the biology of the disease. By 1941, it was well established that exposure of the plant tissue to Agrobacterium causes the Crohn gall disease. Later in 1947, tumor-inducing capacity was proposed by Brown and colleagues. So as per the tumor-inducing principle, it was proposed that the component that could possibly cause the Crohn gall disease may be: one is the metabolic product of the Crohn gall bacterium, that is the Agrobacterium tumefaciens. So possibly Agrobacterium synthesizes some metabolite that can cause the or that could cause the Crohn galls. Second, it is also possible that because of the bacterial infection, some normal constituent of the host plant, it may be converted into tumor-inducing substance or signal and hence result in the Crohn gall disease. Further, it could be a chemical fraction of the bacterium, or it may be some virus or other agent that enters the plant host with the Agrobacterium and causes the disease. Later in 1957, Lauret and colleagues reported the presence of unusual amino acid derivatives, that is the lycopene from the Crohn gall disease. And these amino acid or unusual amino acid derivatives, they are called as the opines. This was the first and a significant step towards understanding the biology of Agrobacterium, that is how the infection takes place. Soon after, it was realized that bacteria utilizes these opines, that is the unusual amino acids, as a carbon source and the nitrogen source for its own growth and development. And different strains of Agrobacterium uses different types of opines. So in later part of 1960s, several developments were reported about the agrobacterium biology, and the concept of plant transformation was identified. By 1970s, technologies were available that could help in identification of the tumor inducing substance. So during late 1960s or 70s, evidences suggested that the host cells are genetically transformed. Bacteria specific opines were produced in the galls. That means, depending upon the type of the bacterial or agrobacterium strain that was causing the disease, different types of opines were synthesized or reported from the tumors. However, same bacterial strain always produced same type of opines. And finally, intimate association of the bacteria and the plant cells was important for the transformation. In 1975, tumor-inducing plasmids were identified. That means it was worked out that it is the Ti plasmid that was responsible for causing the gall disease. And if we transfer this plasmid, that is the Ti plasmid, from a virulent strain of Agrobacterium to an avirulent strain of Agrobacterium, it transformed the avirulent strain into a virulent strain. So it means that the Ti plasmid carried the virulence related signals. Subsequently, several critical questions were addressed, like what is the segment of the DNA that is transferred to the plant genome? Where does it reside? How is it transferred? And what are the different types of components that are involved in this transfer? And later, it was hypothesized that it can be used to transform the plants with the desired DNA segments. In fact, in early 1980s itself, first report of plant transformation with the desired DNA segment using Agrobacterium was published, and since then we have come long way in this field where Agrobacterium is used for as a plant genetic engineering tool. Let's learn about the biology of the Agrobacterium infection, or the mechanism of the Agrobacterium infection. By now, we know that the Agrobacterium naturally transforms the plants during the infection and causes the crown gall disease. And it carries a plasmid DNA in addition to the chromosomal DNA. We also learned that these plasmids, that is the Ti plasmid or the tumor inducing plasmids, are required, or this is essential to cause the transformation event and hence the disease. So presence of Ti plasmid is required for the transformation process. These plasmids are relatively large in size, and it may vary from about 100 kb to up to 1 megabase or even more in some cases. However, the Ti plasmid of the Agrobacterium tumefaciens that is commonly used for the plant transformation is of about 200 kb in size. Now, if we zoom in to see the various components of the Ti plasmid, so here is the structure of a Ti plasmid. Typically, it has all the elements that are present or required in the plasmids for their multiplication and existence. However, I will focus on the major regions that are involved in the infection process. So these two regions, that is the tDNA region and the virulence region. These are the two major components of the TI plasmids that are involved in the infection process. The tDNA or the transfer DNA segments start from the left border and till the right border. It all is a transfer DNA or tDNA. So whatever is present between these left and right borders is called as a tDNA. And second is the virulence region, which code for several genes required to establish the infection in the plants. Earlier we discussed that Agrobacterium transformed the plant cell with its own DNA segment. And this region between the left and right borders is the segment that is actually transferred to the plant genome during the infection and is called as a transfer DNA or a tDNA. Further, this segment can be divided, or it has two major components. One is the oncogenic region and second is the opine synthesis region. So the oncogenic region means that the tDNA region that carry the genes encoding for auxins and cytokinin, that is the plant hormones. These plant hormones, which are synthesized from this tDNA region, they stimulate the plant cell division, and in this case their continuous production results in the uncontrolled growth, which causes the tumors or the tumorous growth. Second component or second part of tDNA is the opine synthesis genes. This region contain a gene that result in the synthesis of opines. Now these opines are the derivatives of amino acids, and they are used by the Agrobacterium as a source of carbon and nitrogen. So basically it's the food for the Agrobacterium. So this whole region from left border to the right border, that is the tDNA region, it varies somewhere from 20 to 30 kb in size in different bacterium strains where TI plasmids are present. Among different Agrobacterium strains, this opine synthesis region may vary, and different strains can synthesize different types of opines. So the TI plasmids are also categorized based upon the type of the opine it synthesizes, like octopine or the nopaline plasmids, as they synthesize octopine or nopalines respectively during the infection in the tumors. Second important region is the virulence region. Now it is about somewhere 30 to 40 kb in size, and it carries at least 6 to 8 operons that code for more than 20 genes. So you know that in prokaryotes genes are organized in operons, that is more than one genes, they are controlled by a single promoter, and therefore these 6 to 8 operons which are present in this virulence region, they synthesizes about 20 to 25 proteins in this case. These proteins are responsible for the whole transformation process, that is the generation of the tDNA and the transfer of this tDNA from the Agrobacterium cells to the plant cells. Now, depending upon the various events during the infection process, this whole infection process can be grouped into 5 or 6 steps. First one is the bacterial colonization, that is the bacteria colonizes on the plant parts like roots or stem. The next is the induction of the virulence genes. So once the bacteria colonizes, it needs to transform the plant cells, and hence it activates the expression of various virulence genes which are required for the infection process. Next step is the tDNA processing, that is, the DNA segment that would be transferred to the plant cell for integration into the plant genome is prepared. It is followed by its transfer, and now the DNA segment is transferred from the Agrobacterium cell to the plant cell. So once it reaches in the plant cell, it needs to be part of the host genome, and hence integration of this tDNA into the plant genome takes place. Finally, when the tDNA is integrated into the host genome, plant is not able to differentiate it from its own DNA, and it is transcribed or expressed as a plant's own component. And ultimately, the products which are coded, or the components which are coded from this tDNA while it is in the as a part of the plant genome, they ultimately causes the disease symptom in the plants and causes the crown gall tumors. Let's learn about these various steps of infection in little more detail. So here is a plant cell, and Agrobacterium should be in the soil. It's a soil bacterium, so they are there in the soil somewhere in the vicinity. And so the first step is that bacterial colonization, and it occurs through a chemotaxis movement. So when there is an injury to the plant releases secondary metabolites as a defense response that probably inhibit the growth of microbes or the pathogens on the wound sites and protect itself. However, Agrobacterium uses this event as an opportunity for the infection. It senses the release of these secondary metabolites and move towards the site of injury through the chemotaxis movements. So the Agrobacterium moves close to the plant cell at the site of injury and attaches itself to the plant cells there. Second step is the induction of virulence genes. The virulence genes are present on the TI plasmid and need to be induced in order to start the infection process. Now, the bacterial membrane has special receptors that are coded from the genes located in the virulence region itself, that is Vir-A gene. So these are the Vir-A receptors. Now these Vir-A receptors, which are present on the membrane of the bacteria, they interact with the plant phenolics or the secondary metabolites which are released by the plant, and this interaction kind of confirms that there is an injury to the plant, and hence there is an opportunity for the infection, and bacteria may initiate the infection process. So basically, this interaction confirms the initiation of the signal cascade for the next step. So these receptors on the interaction with secondary metabolites starts a signal cascade that signals the virulence region on the TI plasmid to induce the transcription from other operons located in the virulence region. The next step is tDNA processing, that is the preparation of the tDNA to be transferred to the plant cell. Now, these virulence gene products that are formed after induction has three major jobs. First is that they activate the synthesis of the tDNA copy. So obviously, the tDNA that we need to transfer to the plant, it need to be duplicated or copied. So these proteins, they make a copy of the tDNA region that need to be transferred. Second job is to protect the newly generated tDNA copy. Now, this newly generated tDNA copy is a single-stranded DNA, and it is prone to degradation from the nucleases. Therefore, some of the proteins which are coded from this virulence region, they make a protective covering on the single-stranded DNA or single-stranded tDNA and make it inaccessible to the nucleases, and therefore nucleases cannot degrade it. In addition, these proteins also help in packaging the tDNA and make it small in size, which makes its transfer to the plant cell much easier. Next step is once the tDNA is prepared, next step is the tDNA transfer. So now the tDNA that has to be transferred is ready for its transfer. But in order to transfer to the plant cell, there should be some communication channel between the two cells. So third point or third job of these proteins is to create a communication channel between the two cells, that is the agrobacterium cell and the plant cells. So while the tDNA is being prepared and packaged, this communication channel is also established. So the other virulence proteins, specifically the Vir-B proteins, these create a type 4 secretion system type of the communication channel which connect the cytoplasm of the two cells, that is the plant cell and the agrobacterium cell. And now this packaged tDNA is recruited or directed to the transfer apparatus, that is this communication channel on the membrane, and some of the proteins encoded from the virulence region perform this transfer process too. The tDNA passes through this pore and reaches in the cytoplasm of the plant cell. Next step is the integration of the tDNA into the plant genome. Now, in order to integrate into the plant genome, the tDNA has to enter into the nucleus of the plant cell, as the nuclear genome is present in the nucleus. So this is a nucleus of the plant cell, and the single-stranded DNA is coated with the proteins and is packaged while it is present in the cytoplasm of the plant cell, as it needs to be protected from the nucleases here too. So it stays in the packaged form in the cytoplasm of the plant cell too. Now, in addition to the role of packaging, these proteins which coats the single-stranded tDNA, they have an additional function, and that is targeting of the DNA to the plant nucleus. So that is, they have the nuclear localization signals on them, and it helps or it directs the tDNA towards the nucleus. During this process, the transfer of tDNA to the nucleus, it takes help from the other plant proteins too. So basically, these agrobacterium components, they interact with the plant proteins or plant components in the plant cell and recruit some of the plant's proteins or the plant's machinery for directing the or transferring the single-stranded tDNA complex to the nucleus of the plant cell. Now the tDNA has to enter into the nucleus, and for this tDNA is recruited or directed to the pores of the nuclear membrane through which it enters into the plant cell nucleus. And this whole process is an active process, that is, it utilizes the energy for the entering into the nuclear pore. Once it enters into the nucleus, it integrated into the plant genome, and this integration is a random event. So it may integrate any part of the genome anywhere. Next step is the expression and development of the crown galls. So once the tDNA is integrated into the plant genome, it behaves as plant's own DNA. So now the plant would not be able to differentiate between the tDNA which comes from the agrobacterium and get integrated into the plant genome, and hence it transcribes the genes present on the tDNA in the same way as its own genes. Now, if you remember the constituents of the tDNA, it had two regions we discussed in the earlier slides. One, it carries the genes for the opine synthesis. So when these are expressed, that is the genes for the opine synthesis are expressed, these result in the synthesis of the opines, which are later used as a carbon or nitrogen source by the agrobacterium. And obviously what kind of the opine they will synthesize, it will depend upon the agrobacterium strain and what kind of gene for opine synthesis that strain carries. Agrobacterium is benefited from this association in the form of these opines which are used as a food, and now it is making plant to make the food for its own growth in the form of the opines. So in addition, tDNA also carries the genes for the plant hormone synthesis in the oncogenic region. So that is the gene encoding for the plant hormones. When they are expressed, they synthesize the auxin and cytokinins. These hormones, when produced inside the cell, they stimulate the cell division or stimulate the plant cell division, and in this way they make more cells. That means more food for the agrobacterium, but at the same time more hormones that keep on stimulating the cell division, which lead to the uncontrolled cell division. So basically this process continues, that is when the tDNA which is integrated into the plant genome, when it is expressed, it makes two components. One is that it makes the opine, so bacteria will be using it as a source for the food and enjoying it and growing it, need not to do anything else. And second is they will keep on synthesizing the hormones which will make the more and more hormones which lead to the uncontrolled cell division. So basically formation of these hormones will stimulate, like if it was infected in one particular cell, formation of these hormones will stimulate the division of cells. However, when these cells they divide into two or they redivide into multiple cells, the tDNA which is integrated into their genome, it is also replicated with these divisions. And all the new cells, though they will be making more and more of the opines, that is the food for the agrobacterium. However, all of these cells at the same time they will be making more and more hormones or the plant hormones which stimulate the plant cell division. That means soon the division of the plant cells becomes uncontrolled. And in this way, uncontrolled synthesis of the plant hormone leads to the uncontrolled division of the cells, leading to the formation of tumors. And this is called as the Crohn-Gall disease. And this is how the agrobacterium infects the plant, transforms the plants, and how smartly it integrates its own DNA into the plant genome, and after that it makes the plants to work for it. That is, it is making the food for the agrobacterium, and agrobacterium is just growing and enjoying by using the opines made by these dividing cells. So in brief, Agrobacterium tumefaciens is a gram-negative bacterium that senses the plant secreted phenolic compounds and move towards injured plant chemotactically. In nature, Agrobacterium usually infects dicots through the open wounds, and during the infection process, it transfers a small segment of its own DNA to the plant cells, where this DNA is integrated into the plant genome. Now, several virulence gene products participate in the synthesis of single-stranded tDNA from the Ti plasmid, and its transfer to the plant cell through the type 4 excretion system. In the plant cells, this tDNA integrates into the plant genome and express the genes that are present on the tDNA. The expression of the genes from the tDNA results in the synthesis of the opines that are used by the agrobacteria as the energy source, and the plant hormones, that is, the auxins and cytokinins, which result in the uncontrolled cell division leading to the tumorous growth. Now, we learned the mechanism of Agrobacterium infection, and the question now is that how can it be used as an engineering tool to transform the plant cells? Now, if you carefully study the whole mechanism of this Agrobacterium infection, you will find that right from the bacterial colonization to the integration of the tDNA into the plant genome, which includes the induction of virulence gene, processing of the transfer transfer of tDNA to the plant cell and its integration into the plant genome. tDNA is not taking part in this whole process. In other words, tDNA do not code for any of the components that are required for these processes, that is, from the colonization till the integration of tDNA into the plant genomes. All of these required components are coded by the Ti plasmid, that is, the virulence genes are performing all these jobs which are involved in this transfer and integration. And there are some other components that are coded by the bacterial genomic DNA that may also help in the infection or make the infection more efficient. Now, the tDNA encoded genes start to work only after the tDNA is integrated into the plant genome, and now they causes the once they are there, they are activated, they are transcribed, they causes the actual disease, that is the crown gall disease, by synthesizing the plant hormones. Therefore, the tDNA encoded components do not have any regulatory role in the transfer from the agrobacterium cell to the plant cells and then to the integration. And this property of agrobacterium tDNA is used to develop it as a plant transformation tool or the plant engineering tool. So this is a typical structure of agrobacterium. It has a chromosomal DNA and the Ti plasmids. During the tDNA transfer process, the products of the virulence genes, they recognizes the border sequences, that is the left border and the right border, and prepare the copy of the DNA present between these borders. So for the whole process, it does not matter what kind of DNA is present between these borders. If the borders are intact, transfer process or the preparation of the tDNA will take place, and transfer will also takes place normally. Therefore, even if we replace this tDNA segment that has auxin, cytokines or opine synthesis genes with any of other DNA segment, non-specific DNA segments, or the DNA segments carrying the genes of interest or the desired genes that we want to transfer to the plant genome, Agrobacterium would not recognize this change as long as left border and right border are intact. And this property basically is used for the engineering or agrobacterium mediator transformation. So this engineered TI plasmid or the recombinant TI plasmid, when it is transformed in the agrobacterium strain after removing the original TI plasmid, so now the agrobacterium has the TI plasmid, but the tDNA of this plasmid doesn't carry the oncogenic endopine synthesis genes, rather it carries the gene of interest. And so these recombinant TI plasmids, they are also called as the disarmed plasmids. Now this agrobacterium strain with the new recombinant TI plasmid can be used for the transformation with the gene of interest or any DNA segment that is placed between these left and right borders, and agrobacterium will transfer this segment to the plant genome, thinking that it is transferring its own DNA for synthesizing the opines, that is the food for himself. Let's have a quick look on the process once again, that is how the tDNA transfer takes place using the recombinant TI plasmids when we have replaced the tDNA region with the gene of interest that we want to transfer to the plants. So here is a plant cell, and we infect the explant with the agrobacterium having engineered disarmed TI plasmid. So the first step is the bacterial colonization, and it occurs through the chemotaxis movement. So when there is an injury to the plant cell will release secondary metabolites as a defense response, and agrobacterium use this event as an opportunity to infect. It senses this release of the secondary metabolites and move towards the site of injury and attaches obviously itself to the plant cells. Second step is the induction of the virulence genes. So now the Vir-A receptors which are present on the membrane, on the bacterial membrane, they will interact with the secondary metabolites, and they will start a cascade of signals to initiate the transcription from the virulence region. So this interaction between the Vir-A receptor and secondary metabolites starts the signals for the induction of transcription from these virulence genes. Next step is the tDNA processing. That is preparation of the tDNA to be transferred to the plant cells. Now these virulence gene products encoded by the virulence gene has again three major jobs, and the first one is to activate the synthesis of a tDNA copy, and where D2 specifically, that is the proteins encoded from the D operon, they play a major role in this case. It creates a single stranded tDNA copy. If you see here, the TI plasmid or the tDNA of this particular plasmid doesn't have the genes encoding for plant hormones or the opines, but still the single stranded tDNA copy is prepared normally, as the genes encoded by the tDNA, they do not have any regulatory role for this whole process of tDNA synthesis and transfer. What it recognizes only the left and right borders. If these two sequences are intact, it will create, or it will make the copy of the DNA or tDNA, whatever is there inside these borders, and that will be transferred. And now the second job is to protect the single stranded tDNA, which is prone to degradation from the nucleases, and therefore some proteins which are encoded again by the virulence region, they protect the single stranded DNA, and the proteins specifically coded from the E operon, they play this role, specifically the Vir-E2 protein. This coating of the DNA certainly protects the single stranded DNA from the nucleases. However, in addition to that, it packages the tDNA into a very small complex, which makes the transfer of the tDNA complex to the plant cell easy through the communication channel which is formed between the two cells. And while the tDNA is being prepared and processed or packaged, where B protein creates a communication channel that connects the bacterial cell to the plant cell. So this communication channel is created by several proteins which are encoded from the operon B. At least 10 or 11 proteins are encoded from this Ver B operon. The packaged tDNA is directed to the communication channel or this T4 secretory system tube, and it is transferred through this channel to the cytoplasm of the plant cells. The next step is the integration of this tDNA into the plant genome. Now the single-stranded tDNA complex has reached in the plant cell cytoplasm. However, in order to integrate into the plant genome, it needs to enter in the nucleus of the plant genome, as the nuclear genome or the plant DNA, genomic DNA, is present inside the nucleus. The Vir proteins that protected the single-stranded DNA or helped to package it into a smaller complex, which helped its easier movement or the translocation through the communication channel. It has an additional function, that is, these proteins has nuclear localization signal. What does that mean? It means that if the signal is present on any component, it tells the machinery or cells machinery that my location is in the nucleus and I should be assisted to reach into the nucleus. And therefore, because of the presence of these nuclear localization signals on the proteins which are associated with the tDNA complex, this tDNA is recruited to the nuclear membrane. And on the nuclear membrane, there are the small pores which are present. Through these pores, this complex enter into the nucleus. And finally, the tDNA integration takes place. That is, this tDNA is integrated into the plant genome, and this integration event is random. So once the tDNA is integrated into the plant genome, next step is the expression of the tDNA segments or the genes present on the plant cell. So after the integration into the plant cell, it becomes the part of plant's own genome, and that means now plant would not be able to distinguish the tDNA that it gained from the Agrobacterium infection or TI plasmid of the Agrobacterium from its own other part of other DNA, and hence the plant's machinery would transcribe and express the component of the tDNA in the same way as its own genes. Now, if you remember the constituents of the tDNA, it had two regions. First one was the opine synthesis genes. That is, when these genes were expressed, they resulted in the synthesis of opines, which were later used by this agrobacterium as a source of carbon and nitrogen. So the agrobacterium was benefited from this association, that now it is making the plant to make food for its own growth. However, now we have replaced the opine synthesis genes with the desired DNA or the genes of our own interest, and hence as the gene of the opine synthesis is absent, opine synthesis will not take place in this case. And similarly, the second part of the DNA, tDNA, or second component of the tDNA had the genes resulted in the synthesis of plant hormones, that is the auxins or the cytokinins. And again, as we have replaced the tDNA with the genes of interest, the genes that coded for the plant hormones, they are not there, and obviously the plant hormones will not be synthesized in this case. So what will it synthesize? It will transcribe the genes that are present on the tDNA that is integrated in the plant genome. That means in this case it will express or synthesize the product of our genes of interest or the trait of our interest for which we had put the genes on the tDNA. So now, though this cell has been transformed by the agrobacterium, however, in the absence of the plant hormone synthesizing genes, it will not be able to cause uncontrolled division or the Crohn-Gall tumors. In this way, Agrobacterium infection process can be used to transfer the DNA segments of our interest to the plant genome. Agrobacterium developed this whole process of transformation for its own growth and survival. However, by disarming the Ti plasmid, we are using it for plant genetic engineering. So basically, we are using the natural capacity of the agrobacterium of plant transformation to transfer the genes of our interest to plant genomes. Now, once the cells have been transformed, these are stimulated to divide on the in vitro culture medium, and for this purpose we use the auxins or cytokinin hormones, but in a more regulated way. These transformed cells divide and form the callus, and usually selection markers like the antibiotic resistance genes are used for this purpose. That means when the tDNA carries a gene segment encoding for an antibiotic resistance gene, only the transformed cells would be able to grow on the in vitro culture medium supplemented with that antibiotic, and untransformed cell would die or would not be able to grow. And hence in this way we can selectively multiply the transformed cells to form the callus, and later they are induced for the morphogenetic responses, and complete transformed plants or the transgenic plants are regenerated. We had studied this process of regeneration and morphogenetic responses or the hormonal control of morphogenetic responses in the first unit. However, we will discuss a little bit more about it, how to characterize the transgenic plants in the next lecture too. So in summary, today we learned about the Agrobacterium tumefacium, the biology of the Agrobacterium, how it causes the Crohn-Gall disease in plants, and how it prepares the tDNA that is transferred into the plant, and where it is integrated into the plant genome, and how from the integration of this tDNA segment opines are synthesized and uncontrolled division is initiated. Finally, we discussed about how can we replace the tDNA from the Ti plasmid, and it still is able to cause the transformation, and so we can transfer the genes of our interest by constructing these recombinant Ti plasmid. And in this way, Agrobacterium can be used as a plant genetic engineering tool. So in the next and the final lecture of this unit, you will learn a little more about the Agrobacterium mediator transformation, especially the vector construction and its applications for the plant genetic engineering. Thank you.